In this podcast, we're going to talk about the glomerulus and filtration. Before we start that, this is just a summary of what we've already said with respect to the kidney. Blood supply defines the boundaries in the kidney, so lobar arteries describe the renal pyramids, the arcuate arteries, and of course veins define the corticomedullary junction. We know the parenchyma reflects the structure of nephrons and collecting ducts in the kidney. Renal corpuscles, proximal and distal convoluted tubules are cortical structures. These are sometimes called the cortical labyrinth. Medullary rays, which are cortical structures, point towards the medulla. These contain the straight segments of proximal and distal tubules and the collecting ducts. And of course the thin limb of Henle's loop and the largest collecting ducts are medullary structures. And we note that these large collecting ducts in the medulla are sometimes called the papillary ducts or ducts of Bellini. This slide is just a summary of what we have already said. Here we've got the lobar or interlobar artery coming up and then branching and defining the arcuate artery. Off of the arcuate artery we get this interlobular artery, also called the cortical radial artery. Remember, it is from the interlobular artery that the afferent arterioles arise, which give rise to the glomerulus, and from the glomerulus you get the efferent arteriole, and remember, from the efferent arteriole you get the capillary beds that surround the nephrons, whether they're the peritubular capillary beds on the short loop nephrons or the vasa recta on the juxtamedullary or long looped nephrons. This slide just diagrams the path that urine takes as it is produced from the glomerulus as it makes its way out into the collecting ducts. And I think you already appreciate this, but one goes into the proximal convoluted tubule, to the proximal straight tubule, to the thin descending limb of Henle's loop, to the thin ascending limb of Henle's loop, to the thick ascending limb of Henle's loop, into the distal convoluted tubule, and then into the connecting tubule and the initial collecting ducts. And from those initial collecting ducts, the urine is going to make its way into the cortical collecting ducts, then in the outer regions of the medulla into the outer medullary collecting ducts, and then into the deeper collecting ducts in the inner medulla, the inner medullary collecting ducts, and finally these all merge and coalesce to form the ducts of Bellini. So we're going to focus for the most of the remainder of this podcast on the structures in the glomerulus. This is a diagrammatic view that we've already looked at of the renal corpuscle. So you can see the afferent arteriole entering the corpuscle, branching and ramifying into the glomerular capillaries, and then coming off of the glomerular capillaries, the efferent arteriole. The diagram shows the capillaries covered with podocytes. We'll talk about those in a few minutes. And you can see a portion of the distal tubule here with the macula densa, and the artist has drawn this purposefully to show a difference in luminal size between the afferent and efferent arteriole. And you might imagine that if you could regulate the luminal diameter of the afferent and efferent arteriole, you might regulate blood flow going through the glomerulus and hence glomerular filtration rate. And here, just the histological image showing the renal corpuscles. You can see the urinary space that I'm highlighting like so. You can see the parietal layer of Bowman's capsule. The visceral layer of Bowman's capsule would be the podocytes that are directly opposed onto the capillary endothelial cells at the light microscopic level. Of course, you can't resolve the podocytes from the endothelial cells. And here you can see the macula densa in this profile region of the distal convoluted tubule, and then you can see profiles of the proximal convoluted tubule. They're these darker, more eosinophilic staining tubules. So this is a diagrammatic view of the glomerular filtration barrier. It's a tripartite structure. and it consists of the endothelium that make up the glomerular capillaries, and the podocytes, which make up the visceral layer of Bowman's capsule, and the basal lamina, the common glomerular basement membrane, which is synthesized by both the endothelial cells and the podocytes. And so the diagram here shows in yellow the podocytes, and then the red profiles would be the profiles of the glomerular capillary endothelial cells, and then an intraglomerular mesangial cell. And this greenish structure that I'm highlighting with the pointer would represent the glomerular filtration barrier. The blow-up shows the endothelial cells in diagrammatic form. Note that they're fenestrated endothelium. Here is the common basal lamina, shown like this in the blue. Here are an example of the podocytes, and notice the podocytes have primary processes and secondary processes, and these secondary processes wrap around the endothelial cells, and they almost form like a sieve or filtration slits that play a role in part of the filtration. And then there is what's called the filtration slit diaphragm, a proteinaceous structure between these secondary foot processes, and the secondary foot processes are also given the name pedicles. Just a higher power view of the diagram that we just saw, so you can clearly see the podocytes, you can see the glomerular basement membrane, which is the actual filtration barrier, and then the endothelial cells of the glomerular capillaries. Note that they're fenestrated, so you can see that like so, or you can see the fenestrations here, and then an intraglomerular mesangial cell, and then the sawtooth-like appearance in the diagram is just meant to represent the secondary foot processes, or the pedicles. We've just said the capillaries are fenestrated. They do not have diaphragms. You can see the intraglomerular mesangial cells here, and interglomerular mesangial cells play an important role in not only supporting the filtration barrier, but also, as we suggested in an earlier podcast, in cleaning the filtration barrier. As we said, the podocytes exhibit primary and secondary foot processes. The pedicles have a well-developed glycocalyx that consists of a variety of negatively charged proteins that are collectively called podocalyxins. Here is an electron micrograph, both at the scanning electron microscopic level and at the transmission electron microscopic level. So at the scanning level, the parietal layer of Bowman's capsule has been peeled away, and so you're looking down onto the surface of the glomerulus, and you can see the podocytes with their primary and secondary foot processes. The transmission electron microscope is showing this at a higher magnification, and the structure consists basically, the basal lamina, of a lamina rara externa, which is synthesized by the podocytes, a lamina rara interna, which is synthesized by the endothelium, and then a lamina densa. So on the transmission micrograph, these are secondary foot processes of the podocytes. Here would be the lumen of the glomerular capillaries with profiles of the endothelial cells, and note that these endothelium are fenestrated. Exactly how the glomerular basement membrane contributes to macromolecular filtration is not clearly understood. What we do know is that the lamina rara, which I'm going to show here like this and here like this, and I'll label them for you in just a second. The lamina rara is an electrostatic barrier, and it's due to proteins in that barrier like heparan sulfate that restrict the passage of negatively charged molecules. The lamina densa, which I'm highlighting here, and again I will label it in just a second or two, is a physical barrier due primarily to collagen type 4, and the lamina densa restricts movement of molecules that are larger than approximately 70,000 Daltons. Here, the lamina densa, this electron-dense material between the podocyte foot processes and the profiles of the glomerular endothelial cells. Here is the lamina rara externa, this more electron-lucent area synthesized by the podocytes. The lamina rara interna, the electron-lucent area on the capillary side of the lamina densa, the lamina rara interna, of course, synthesized by the endothelial cells. And here is the slit diaphragm. This slit diaphragm probably plays the most important role in determining the molecular sieving characteristics of the glomerulus. The slit diaphragm contains a variety of podocalyxins. I'm not going to expect you to remember these for an exam, but some of these podocalyxins would be like nephrin, Neph1 and Neph2, FAT1, FAT2, podocin, CD2AP, and zonula occludens 1. These proteins also interact with cytoskeletal proteins within the podocytes, and there's an interesting review article for you that's on the D2L site in the New England Journal of Medicine, and this article is suggesting that indeed the interactions of macromolecules with this filtration slit diaphragm probably plays the most significant role in filtration at the glomerulus. The JG apparatus, just to review for you, consists of the JG cells, which secrete renin and other substances in response to blood pressure. The JG cells shown here in the tunica media of the afferent and efferent arteriole, the macula densa cells in the distal convoluted tubule, thought to monitor osmolarity of the urine and will of course signal the JG cells to secrete renin and other hormones. There are nerve fibers which are not shown in this cartoon that are thought to stimulate the JG cells. These nerve fibers are beta-adrenergic fibers. There are the extraglomerular mesangial cells. Function of these cells not really known. They may play some supporting role here. And then the intraglomerular mesangial cells shown here. These are from smooth muscle. They're modified smooth muscle cells. They support the glomerulus. They clean the glomerular basement membrane, and they regulate glomerular blood flow and filtration. And here again, both the extraglomerular and intraglomerular mesangial cells as modified smooth muscle cells may play some role in tubular glomerular feedback and regulating glomerular filtration rate by their ability to contract. They may regulate how much blood flows through the afferent or efferent arteriole. Control of blood pressure by the kidneys, as we've already suggested, the JG cells secrete renin, and this causes an increase in blood pressure because of vasoconstriction. The JG cells may secrete angiotensins and angiotensin-converting enzymes to play a role in tubular glomerular feedback. As we said, the JG cells are modified smooth muscle cells. When blood pressure increases, they are thought to stretch, and that stretching may down-regulate their secretion of renin.